Hello, everyone. Welcome and thank you for taking part in MDPI's webinar entitled Transporters for Organic Cations. My name is Mina and I'll be your host for today. This is the second webinar in the International Journal of Molecular Sciences series based on a special issue from our journal of the same name. If you're interested in contributing to this topic, don't hesitate to submit your research. Please check the webinar's webpage for more information. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's webpage. Today's session is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us live. Certificates of attendance are available upon request. To get yours, please send an email to ijms.webinar@mdpi.com. Certificates will be delivered to those who have registered and are watching on Zoom, not on YouTube. Thank you for your understanding. There will be 4 presentations today of 20 minutes each. The presentations are followed by a question time moderated by the chair. If you have any questions for our speakers, please post them in the Q&A box at the bottom of your screen. You can see it at the bottom of the Zoom screen. Please also remember to mention the name of the person to whom you are addressing your question. If you want to ask the question yourself, you can also raise your hand during the question time and I will hand you the mic. Today's webinar is being chaired by Professor Dr. Giuliano Ciarimboli from the University Hospital Münster in Germany. Giuliano Ciarimboli studied biology at Pisa University in Italy and was a scholarship holder at the Institute of Clinical Physiology of the Italian Research Council in Pisa from 1989 to 1994. During this time, he was involved in the study of endogenous digitalis-like factors and of pulmonary metabolism of basic amides. From 1995 to 1999, he worked in the laboratory of Professor Stolte at the Hanover Medical School in Germany and investigated the role of charge, dimension, and conformation of proteins in their glomerular filtration. 1999, the University of Hanover in Germany awarded him a PhD degree in the natural sciences. And in 2001, he joined the group of Professor Schlatter in experimental nephrology at the Münster University Clinic in Germany. Dr. Ciarimboli currently holds the position as professor at Universität Klinikum Münster in Münster, Germany. His research interests include regulation of organic cation transporters and their interaction with drugs. Thank you, Giuliano, for chairing today's webinar. That's it for me. I now hand over the mic to our chair, and I hope you all enjoy the webinar.
Thank you very much, Mina, for the kind introduction. And I would like to welcome everybody to this webinar and to present my co-speakers that are Professor Lynette Daws from the University of San Antonio in Texas, and Professor Cesare Indiveri from the University of Calabria in Italy, and Dr. Michele Visentin from the University of Zurich in Switzerland. This is a webinar to present the new special issue of the International Journal of Molecular Sciences, that is Physiology, Biochemistry, and Pharmacology of Transporter for Organic Cations. You can see that there is a number, this is the 2. That means that we have already the first volume of this special issue that was released already last year. And we had a good success, I think, because we had 14 contributions from 61 authors. And this contribution were divided in 11 research papers and 3 reviews. The submission closed in July, And today we have almost 10,000 cumulative views for the publication in this issue with 47 cumulative citations. So I think it was rather a success, this issue. So I would like to present you general physiological and pharmacological properties of transporter for organic cations. As Mina said, I'm working in Münster. You can see here on the left bottom a picture of a town. This is a very nice town in northwest Germany. And on the bottom, but on the right, you can see a picture of a university clinic here in Münster where I'm working. This slide shows a schematic representation of a plasma membrane. The development of the plasma membrane was an important achievement in the evolution of cellular life, because a plasma membrane can protect the genetic material, the DNA or RNA at the beginning, from the influence of the environment. But at the same time, this plasma membrane was a problem for exchange and communication between the new formed intracellular space and the extracellular space, because only hydrophobic molecules can move across the plasma membrane without any problem, but hydrophilic molecules cannot pass the plasma membrane. For this reason, the cells adapted and embedded in the plasma membrane many different transport proteins. Here, as you can see, we know now that around 800 human genes are coding for transport protein, and these genes are 5% of total. So transporter are very important proteins for the cells. And when we are trying to classify the transporter, there is a division between transporter of the solute carrier series and other transport proteins. Among the other transport proteins, we find water channels, ion channels, and transporter that need dielectric energy in form of ATP hydrolysis, such as pump and the ABC transporter. The other transporter that are 50% of the whole transporter belong to this solute carrier series, and here we can find several different types of transporter that are working as a coupled transporter. That means that they can transport two substances, or as exchanger, they are transported two substances in different direction, or they are passive transporter. This transporter can be localized on the plasma membrane, but also on the membrane of organelles of the cells and also on the membrane of the vesicular structures in the cells. So these transporters together are very important to maintain the cellular homeostasis, and so they are very critical for life. Going back to this solute carrier transporter, there is a classification based on the homology of amino acid sequences between the transporter, and according to this classification, we have identified 65 different families of SLC transporters. Among these families, there are few that are transported organic cations that are the substrate we are interested into. And in this presentation, I will focus on these two families, the solute carrier 22 organic cation anion zwitterion transporter family, and the family SLC 47 multi-drug and toxin extrusion family. When we try to see the evolutionary relationship between these transporters, we can calculate how similar are the nucleotide sequences. And the distance along branches is inversely related to degree of sequence identity. So we can find that in the SLC22 family, there are 3 transporters that are very close, and they have organic cation transporter 1, 2, and 3. And the H means that they are human transporter in this example. And in the same family, we find 2 other transporters that are closely related. They have OCTN1 and the OCTN2. Not so related to this transporter, but not so far away, we can find the mate family, the SLC47 family, with mate 1 and mate 2K. So the transporters are a little bit different also regarding the secondary structure. As you can see here on the top of the figure, we have a schematic representation of the secondary structure of OCT and OCTNs. These transporters have 12 transmembrane domain. They have both amino and the carboxy terminus in the cell. And they possess a big extracellular loop with glycosylation sites and a big intracellular loop, which have many potential phosphorylation sites that can indicate a possibility to regulate the activity of these transporters. The mate transporter has a rather different structure. Here we can identify 13 transmembrane domain. We have no big extracellular loop, only an intracellular loop that is relatively big. The amino terminus is intracellular localized, and the carboxy terminus is localized toward the extracellular space. So there are some differences between these transporters. But interestingly, this transporter share many substrates. Here you can see a short list of substrate of OCTs, OCTNs, and also of mate. And as you can see, we have substrate that are of endogenous origin and other substrate that are of exogenous origin. The normal properties of these organic cations is that they are positively charged at physiological pH. As you can see, among the substrate of endogenous origin, we can find important neurotransmitters such as acetylcholine, dopamine, histamine, and serotonin, suggesting that this transporter may have an important physiological meaning. But at the same time, we find among the exogenous substrate of the exogenous origin, many important drugs, such as cimetidine, that is an antihistaminic, quinine, that is an anti-malarial drug, metformin, that is an important anti-diabetic medication, and procainamide, that is an antiarrhythmic medication. And I would like to underline at this place that up to 40% of prescribed drugs are organic cations. So this transporter may have also an important pharmacological meaning. I would like to show this interesting slide where you see at first many transporters, and I would like to focus here on the transporter of SLC22 and SLC47 family that are the OCT2, 3, OCTN1 and OCT2, OCTN2, and the mate transporter, mate 1 and mate 2K. On the other side of the slide, you see other transporter, but they are organic anion transporter. But the interesting thing to observe in this slide is that the OCTs, of course, are able to transport substances that are organic cation, as of the blue dots are organic cations, but they accept as a substrate also substances that are without any charge, as of the gray dots are these uncharged organic molecules. it's of high interest to observe that in few cases there is also an interaction of organic anion, as a negative charge substances, with OCTs. So these transporters are accepting many different substances as a substrate. But then I would like to show you another interesting properties of this transporter. You can see this blue dots is a substrate of OCT2, but it's also a substrate of OCT3 and of MATE2K and of MATE1. And probably we can also find, when we are searching among these dots, also substrates that are common to the OCTs and MATE and OCTNs transporters. And sometimes also with organic anion transporter. So that it seems that this transporter have an important characteristic, that is they are polyspecific. They can accept many different substances as a substrate. And it's very interesting to think about the reason for such a polyspecificity. Probably that is due to the special structure of the binding pocket of this transporter. As you can see here, I tried to make a model of this binding site. We can imagine that these transporters have a big binding pocket, and in this binding pocket we can find partially overlapping binding sites for different substrates. So we can imagine that a substrate have a binding site and the substrate 2 as another binding site, but the two binding sites can overlap. For this reason, when we are using different test substances to measure the transport kinetic of this transporter, we can obtain also different numbers regarding the affinity, for example. But the group of Professor Köpsel has widened this hypothesis. He published 2 years ago a work where he could show that the red organic cation transporter 1 contains 3 binding sites for the substrate 1-methyl-4-phenylpyridinium per monomer of the transporter. So the situation is really complicated, and this is an important topic to further investigate for this transporter. Where we can find this transporter in our body, as this slide refers to the situation in humans. When we start from the bottom left here, we have first the kidneys, and the cells of the kidneys are polarized cells. That means that they have 2 different plasma membrane domain, basolateral membrane domain, that is the plasma membrane domain that is facing the blood, and then apical or luminal membrane domain that is facing the urine. As you can see, we can find the OCT2 in humans that is expressed on the basolateral side of the cells, and of the apical sides we can find other transporters that can mediate the excretion of organic cations into the urine. These transporters are the MATE1, the MATE2K, and the OCTN1 and the OCTN2 transporter. Here it's important to say that the driving force for the uptake of organic cations by OCT2 is the electrochemical gradient of the substrate. On the other side, the driving force for excretion of these organic cations that have been taken up into the cells by OCT2 is given by the slightly acidic pH of the urine in this part of the kidney, so that these transporters are working by exchanging organic cations against protons. And this is possible because of these pH differences. In the liver, we find also polarized distribution of this transporter. Here you can see a schematic representation of an hepatocyte, and we can find the OCT1 in human that is expressed on the basolateral membrane of the hepatocytes, that is also called sinusoidal membrane. And on the apical membrane, we can find the mate transporter that are mediating the excretion of organic cations into the bile. In the nervous system, we know that several OCT are expressed. Hopefully, Lynette will tell us something more precise about this. And probably both OCT2 and OCT3 are expressed in postsynaptic neurons and also probably in astrocytes. We can find the organic cation transporter also in the lung. You can see here in the alveolar epithelium, we find the OCTN1. On the apical side of these epithelial cells, we can find also the OCTN2 and the OCT1 and 2, and also the 3 are all the transporters. In the intestine, we can find also the OCT1 that is localized on the basolateral membrane domain of enterocytes. So that we can observe that generally the organic cation transporter are localized on the basolateral membrane domain, while the mate transporter and the OCTNs transporter are localized on apical membrane domain. But it's not always the same. In the lung, the situation is a little bit different. We cannot find such a difference in the apical or basolateral membrane distribution of a transporter, suggesting that the indication of where the transporter has to go after the biosynthesis is not contained in the sequence, in the amino acid sequence of the transporter. When we are thinking of the physiological role of this transport system, I think that we can say that the role of axis OCTs and mate transporter is a clearance role of organic cations. So we can think of the control of neurotransmitter extracellular concentration, for example, in the brain, but also in organs such as the kidney and the liver, in the excretion of potentially dangerous substances. There is an interesting hypothesis about the physiological function of these transporters. This is the remote sensing and signaling hypothesis that is saying that the transporter of the SLC22 family, together with other SLC transporters and also ABC transporter, are part of a large interorgan and interorganism small molecule communication network that maintains homeostasis in epithelial and non-epithelial tissues and body fluid compartments, such as blood, bile, cerebrospinal fluid, and urine. So that this interorganismal communication can result between organisms of the same species, for example, between the mother and the fetus, but also switching organisms of different species. We can imagine that when an animal, a cat or a dog is urinating, another animal can take up these odorant proteins that are distributed into the urine by the action of a transporter. In the olfactory system, there are other transporter that can take up these odorants, and so they can be interpreted by other animals as a signal. Also. I would like now to show you a little bit more about this transporter in the kidney. And here you can see a schematic representation of the kidney with a nephron, a schematic nephron that is the functional unit of the kidney. You know that the kidney, the principal function of the kidney is to filter the blood. And this filter function takes place in a structure that is called glomerulus. And when a tubular system is collecting the filtrate, and is modifying the filtrate before urine excretion. Also with a reabsorption process, the tubulus can save many important substances that are filtrated from the blood, such as glucose, for example. And this action is mediated by transporter. But there is a special function of the kidney that is the secretion of substances into the urine. And this is the secretory clearance of the kidney. And this secretory clearance is mediated by organic cation transporter and by organic anion transporter, because the substances that are secreted in the kidney are of ionic nature. And here you can see more precisely the transport system for organic cation in the proximal tubulus of the kidney. As I already said, we can find on the basolateral membrane the OCT transporter that are taken up organic cations, and these organic cations are then excreted into the urine by the action of several other transporters, among them the MATE transporter and the OCTNs transporter. So that the physiological function is able to secrete in the urine potentially dangerous substances, also metabolism products such as creatinine. Creatinine is a substrate of this secretion axis. But there are many studies that are showing that drugs such as cisplatin and oxaliplatin and metformin are also substrate of this transporter. I would like to show you very briefly some results with cisplatin. So cisplatin has been demonstrated to be secreted into the urine by the action of OCT2 expressed on the basolateral membrane domain of proximal tubular cells, and then the secretion to the urine is mediated by the MATE1 transporter. But in the passage through the cells, cisplatin is also dangerous for these cells and can cause a nephrotoxicity in the proximal tubules. But we know now that the same transporter, OCT2, is also expressed in the cells of the Corti organ and of the cochlea. You can see here a Corti organ of the ear, where hearing takes place. Also, we have hair cells here that express the OCT2. This green labeling is showing the expression of OCT2. But OCT2 is also expressed in the stria vascularis cells that are mediating the composition of the endolymph in this part of the ear. It's also been demonstrated that OCT2 is also expressed in dorsal root ganglia, because it's known that cisplatin can also cause ear and neurotoxicity, and here it seems to be expressed rather in satellite cells than in neurons. In cancer cells, there are few indications of expression of OCT2 in cancer cells, so that the anti-cancer action of cisplatin is probably not due to a transport in cancer cells by OCT2. Interestingly, by competing with uptake of cisplatin by OCT2 with another organic cations, such as cimetidine, it was possible to suppress the ototoxicity of cisplatin and also the peripheral neurotoxicity of cisplatin, without changing the toxicity of cisplatin against cancer cells. But this approach was not so successful for the kidney, as we cannot observe any strong protection of kidney cells against cisplatin, probably because cimetidine is also inhibiting the MATE1, and this inhibition is much more stronger than the one against OCT2, so that the secretion of cisplatin into the urine is compromised. I would like to say that this transporter has an important physiological and pharmacological function. I think you are convinced of this, but there are many situations where this transporter can change their expression and their function. And so the physiology and pharmacology of the transporter have been influenced. For example, by mutations, it is known that these transporters are polymorphic, they have many SNPs, and these SNPs can change transporter characteristics. Or there is epigenetic modification of the transporter. For example, the methylation of the SLC22A1 reduces its expression, so it will influence its function. In animals, it's known that the transporter expression is sometimes depending on the sex of the animals. Also, this has been observed in the kidney of mice and rats for OCT2, but there is no indication that this is the same in humans. Then this transporter can be changed in their expression by pathology. For example, it seems that diabetes mellitus is decreasing the expression of renal OCT2 in rats. And then the function of this transporter is subjected to a rapid regulation by several signaling pathways, for example, by PKC activation, or by interaction with other proteins. For example, the interaction of OCT2 with the tetraspanin CD63 is determining the expression of the transporter in the basolateral membrane. So I think I am ready now. I see no questions, so I would like to introduce Lynette Daws. Dr. Lynette Daws is currently the Frostbank Distinguished Professor in Biomedical Sciences in the Departments of Cellular and Integrative Physiology and Pharmacology at the University of Texas at San Antonio in the United States. Dr. Daws is interested in the role of organic cation transporters in maintaining monoamine homeostasis in brain. She applies primarily in vivo electrochemical, high-speed chronoamperometry, ex vivo neurochemical and behavioral methods to her research, using a combination of genetic and pharmacological approaches. Dr. Daws and her collaborators have revealed organic cation transporter-free. This is a very important result of the group as a potential target to improve therapeutic intervention for the treatment of depression and as a site of action for amphetamine and the related psychostimulant. And I'm very happy that she accepted the invitation to present her work here. The title of the presentation is Organic Cation Transport and Psychiatric and Substance Use Disorders.
Thank you very much, Giuliano, for inviting me to participate in this webinar. And thank you very much for setting the stage so nicely, giving that introduction of organic cation transporters. So good morning, good afternoon, good evening to everyone. Thank you for participating in this webinar. And as Giuliano just said, I'm going to now narrow the focus on organic cation transporters to the brain, and in particular, how they may be involved in psychiatric and substance use disorders. So I'm going to specifically be focusing on organic cation transporter 3, although I'll mention organic cation transporters 1 and 2, and also the plasma membrane monoamine transporter, or PMAT. This player is actually the newest member of the organic cation transporter family, which was cloned relatively recently, just in 2007 by Joanne Wang's group. I want you to keep in mind that all 4 of these transporters are inhibited by dysinium-22, which I'll be mentioning as I go through my talk. And they're all bidirectional transporters for monoamines, including dopamine, norepinephrine, and serotonin, albeit with varying affinities for these monoamines. So my lab has generally been interested in this broad question, which is, do organic cation transporters and or the plasma membrane monoamine transporter contribute to monoamine homeostasis in the brain? This has been a very understudied subject, and it's really only been the last decade and a half or so that people have been really addressing this question. And specifically, I'm going to talk today about 2 examples in particular, which are the consequences for antidepressant efficacy and for the actions of amphetamine. But before I get started on that, just briefly a little bit about where these transporters are distributed in brain. So what you're looking at here is in situ hybridization to mRNA for OCT3 on the left here, OCT2 on the right, and OCT1 on the bottom. And this is work from Sophie Galtron's group. In fact, her group has really been the pioneers, along with Paul Gasser, in defining the expression of these transporters in brain. And rather than dissect this in detail, the big take-home messages are that OCT3 and PMAT, which is not shown here, but take my word for it, OCT3 and PMAT are the most densely expressed transporters in the brain of the organic cation transporters in the brain, and to a lesser extent OCT3. And the important thing to note is that these transporters are positioned in limbic brain regions, important for the regulation of mood and reward, as are the regions that are activated following substance use administration. So we have, for example, the dorsal raphe, the caudate putamen, the nucleus accumbens, various areas of the cortex, as well as hippocampus. Here's the dentate gyrus, showing a relatively rich expression of OCT3. OCT2 is more confined to the cerebellum, as well as blood-brain barrier regions. Whereas in contrast, OCT1 is relatively sparsely expressed in brain, showing up mostly in the cerebellum. In P here is just showing you sense findings and antisense binding to the different OCT isoforms. And as you can see, sense binding is a nice control showing no binding. So what really do I mean when I'm talking about uptake 1 and uptake 2, which is what I'm going to be talking about a lot this morning in my time. So uptake 1, these are the dopamine, norepinephrine, and serotonin transporters, and these are the high affinity transporters for these monoamines. By that, it really means that when these monoamines are around in extracellular fluid in relatively low concentrations, they're very efficiently taken up by these uptake 1 transporters. However, when the concentrations of extracellular fluid increase, they actually have a very low capacity to transport these monoamines, and they become actually very ineffective in clearing them. In contrast, these uptake 2 transporters, or our organic cation transporters and plasma membrane monoamine transporters, they're not at all efficient in taking up these monoamines when they're around at low concentrations, because they have a low affinity for these monoamines. However, when extracellular levels of monoamines get high, these transporters you would expect to become quite engaged, because they have a very high capacity to transport these monoamines. But really, no one's been thinking about this terribly much up until the last decade or so. And it's probably largely driven by the fact that we haven't really considered that the levels of extracellular monoamines would rise to a concentration that would engage these low affinity, high capacity transporters. But we now know that the synaptic concentrations of monoamines reach the millimole range, meaning by the time they spill out of the synapse into the extracellular domain, which is where these transporters are located, both uptake 1 and uptake 2 transporters are located extrasynaptically. These transporters are going to be seeing, if you will, concentrations of neurotransmitter that are in the micromolar range. So it becomes quite feasible that these uptake 2 transporters may play a predominant role in monoamine homeostasis in brain. So this leads to the question then, is that maybe these transporters can help answer the question of why drugs targeting uptake 1 transporters, certainly in DAT, lacking in therapeutic efficacy to treat psychiatric and stimulant use disorders. I'm going to start by talking about antidepressants. So selective serotonin reuptake inhibitors, or SSRIs, are the most commonly prescribed antidepressant currently. Yet they lack therapeutic efficacy for the majority of patients taking these antidepressants. So can these uptake 2 transporters account for this poor therapeutic efficacy of SSRIs? We approach that question in many, many ways, but since we only have a relatively short period of time, I'm I'm going to show you what I think is one of the neatest ways that we showed that yes, they may indeed be involved. So what we did is used in vivo chronoamperometry, which is an electrochemical recording technique that allows us to measure the clearance of these monoamines in the brain of living rats and mice in real time. So what we can do then is couple our carbon fiber recording electrode with a multi-barrel pipette, and we can load that with serotonin and apply or any monoamine and apply in a very controlled manner the amount of serotonin we're delivering to the extracellular space. So what we did was varied the concentration of serotonin to be either low at 0.5 micromolar or 5 times higher at 2.5 micromolar, and then ask the question, how well does a selective serotonin reuptake inhibitor work to inhibit serotonin uptake under these two different conditions of serotonin concentration? On the y-axis here, looking at change in the TAD clearance time, or just the clearance time for serotonin. And what you can see is when we give fluvoxamine, which is like, it's similar to fluoxetine or Prozac, it's an SSRI. When the serotonin concentration is low, we get very robust inhibition of serotonin clearance, as you would expect, by blocking the serotonin transporter. But when the serotonin concentrations were elevated 5-fold, now the SSRI is having no effect at all. And we were quite excited by these findings because there's a very common polymorphism in the serotonin transporter that confers low serotonin transporter expression in individuals harboring that polymorphism. And based on murine studies, we would expect these people to have much higher levels, about 5-fold higher levels of extracellular serotonin just basally. So this and the carriers of that polymorphism are less effectively treated by SSRIs than those without polymorphism. So this just the concentration differential of basal serotonin could perhaps account for that special lack of therapeutic efficacy in these patients. If we then did the same experiment but gave Dicinium-22, the blocker of OCTs and PMAT, we get the opposite. So consistent with OCTs and PMAT being low affinity, blocking these transporters had no impact on serotonin clearance when the concentrations of serotonin were low. But look at this robust inhibition of serotonin clearance when we increase serotonin 5-fold, even greater inhibition than fluvoxamine by itself. And here's just the vehicle to show that it has no effect on serotonin clearance. And these data are published in this particular paper. And I'm just going to wrap up our antidepressant story very quickly because we've published quite a bit on this. This PNAS paper showed that mice heterozygote for the serotonin transporter had elevated levels of organic cation transporter 3, and Dicinium-22 in those animals can produce antidepressant-like effects by themselves, suggesting that perhaps organic cation transporter 3 might be a good therapeutic target for low-expressing serotonin transporter gene variants. We also showed that Dicinium-22 could enhance the antidepressant effect of a selective serotonin reuptake inhibitor. And this just summarizes a lot of these findings where we found that the ability of a drug or drug combination to inhibit serotonin clearance correlated very nicely and very positively with the ability of that drug or drug combination to produce antidepressant-like effects. And most recently, we showed that these findings generalized also to norepinephrine-acting drugs, where here you can see that the effect of desipramine, a blocker of the norepinephrine transporter, was enhanced by co-administration of Dicinium-22. So in conclusion, for the antidepressant part of my story today, blockade of Dicinium-22 sensitive transporters, putatively OCT3 and PMAT, and I say that based on our studies in OCT3 and PMAT knockout mice, which I don't have time to talk to you about today. But blockade of these transporters enhances the antidepressant-like effect of serotonin and norepinephrine transporter blockers and has standalone antidepressant-like effects in mice with genetically low serotonin transporter expression. So this may help explain why the therapeutic efficacy of SSRIs and other classes of antidepressants is suboptimal for many patients. Indeed, there's clinical evidence that suggests that triple uptake blockers, which block the serotonin, norepinephrine, and dopamine transporter, are the most efficacious. And what I haven't mentioned is that OCTs and PMAT take up all three of these monoamines, albeit with different affinities. But when you block one or more of these organic cation transporters, you would expect to get an increase in all three monoamines and not just one. So this could be another reason adding to the excellent, what seems to be an excellent antidepressant-like effect of blocking organic cation transporters. So targeting these transporters may be a really good road for future investigations to develop antidepressants with improved therapeutic efficacy. So switching gears, just to finish up, I'm going to talk a little bit about amphetamine. Amphetamine, as we know, is a psychostimulant. It is used therapeutically for the treatment of ADHD, predominantly in people ages 4 to 17. And there's an increasing problem with addiction related to Adderall treatment. Amphetamine is also abused, and it's known as speed for its stimulant effects. And there's also a growing problem with abuse of new synthetic designer amphetamine-like drugs, also known as bath salts, and the taking of drug combos, and I'll expand on that a bit more in a moment. So how does amphetamine work? Its primary target is the dopamine transporter. So amphetamine, these yellow dots here, enter the dopamine neuron via either the dopamine transporter or diffusion across the plasma membrane due to its lipophilic nature. Once inside, it disrupts the vesicular monoamine transporter to cause the release of dopamine into the cytosolic compartment, where it's then transported by reverse transport out of the dopamine transporter into the extracellular fluid and serves to increase the levels of dopamine. Cocaine, unlike amphetamine, is not a substrate for the dopamine transporter, it's a blocker. So when you have cocaine on board, you would expect to block the effects of amphetamine. So herein lies the conundrum. How can taking combos, so this is taking a DAT blocker like cocaine or MDPV, new synthetic cathinone, with a DAT substrate like amphetamine or methadone, one of the new synthetic cathinones, how can taking these together produce a greater high than either drug taken alone? Also, another part of the conundrum is because amphetamine and its related stimulants act at DAT-net and SERT, they're obvious targets for the development of therapeutics to treat stimulant abuse, but efforts in that regard have been fairly futile to date, with drugs targeting these transporters not being effective in treating psychostimulant addiction. Why could that be? Is there perhaps another mechanism of action for amphetamine and amphetamine-like stimulants that we don't yet know? So we published a paper now a couple of years ago, working with Felix Mayer in Harold Fitte's lab, a graduate student then, now a postdoc in Randy Blakely's lab. And we showed in this paper that OCT3 is in fact present on dopamine neurons, and that amphetamine is not a substrate for OCT3. And what I'm going to show you now is our in vivo data that shows that OCT3 plays a role in amphetamine-induced dopamine efflux in vivo, and that it's also important for the locomotor stimulant effects of amphetamine. So again, we used our in vivo electrochemistry. Just very quickly, we load amphetamine into a molded barrel pipette. We pressure inject it, in this case, into the dorsal striatum, which is very rich in dopamine transporters, as well as organic cation transporter 3. And the amphetamine will cause the release of dopamine. And we can measure that released dopamine by the current generated at the carbon fiber electrode and convert that into a concentration using a calibration factor that we determined in vitro. And we found that we could inject amphetamine at 45-minute intervals and get very nice reproducible signals like this, allowing us to detect the effects of cocaine and Decenium-22 on amphetamine-evoked dopamine release. So what did we find? In wild-type mice, OCT3 wild-type mice, when we give amphetamine, in the black is our amphetamine-evoked dopamine release signal, and superimposed is what happens to that signal when we give artificial cerebral spinal vehicle, which is absolutely nothing, as you would expect. However, when we give Decenium-22, we get nice attenuation of amphetamine-evoked dopamine release. Summary data shown over here on the right. When we give cocaine, we also get the expected attenuation of amphetamine-evoked dopamine release. This is a concentration of cocaine, that fully blocks DAT. So it's interesting to note that we're not getting anywhere near full blockade of amphetamine-evoked dopamine release. And when we give Dicenium-22 and cocaine together, we get an even further reduction in the ability of amphetamine to evoke dopamine release. So Dicenium-22 certainly inhibits amphetamine-evoked dopamine release, but through which transporter is it doing this? So we turn to OCT3 knockout mice. Again, in the control situation, when we give ACSF, you can see there's no effect on amphetamine-evoked dopamine release. Now, unlike in our wild-type mice, Dicenium-22 is without effect to inhibit amphetamine-evoked dopamine release. Cocaine, as you would expect, since these mice have their full complement of DAT, continues to inhibit amphetamine-evoked dopamine release, but when given together with D22, doesn't further inhibit the ability of amphetamine to evoke dopamine release, suggesting that this effect of Dicenium-22 is occurring via OCT3. To see if this made any difference to the behaving animal, we performed similar experiments looking at amphetamine-evoked hyperlocomotion. So here's our wild-type animal. You can see at a dose of 3.2 milligrams per kilogram of amphetamine, we get nice hyperlocomotion. When we pre-treat the animal with Dicenium-22, we get almost complete blockade, which is quite incredible. When we saw this, I was amazed because it suggests that a lot of the amphetamine-induced hyperlocomotion is more mediated perhaps by OCT3 than the dopamine transporter. In our OCT3 knockout mice, amphetamine also stimulated an equivalent amount of hyperlocomotion, which we weren't necessarily surprised by because this is a constitutive knockout, so there's likely compensation in the dopamine transporter or other transporters. But the exciting thing to us was when we gave the OCT3 knockout mice Dicenium-22, now there's absolutely no loss of the effect of amphetamine to promote hyperactivity. So again, it looks like this effect of amphetamine on activity is OCT3 dependent. So just to quickly finish up, I know I'm about a minute over right now. What do I think is going on, or what do we think is going on? As I showed you before, we have amphetamine entering the neuron by both the dopamine transporter and diffusion across the plasma membrane, leading to release of dopamine via reverse transport through the dopamine transporter. Our data suggests that there's another player involved in the actions of amphetamine, and it looks to be organic cation transporter 3. So when we block the dopamine transporter with either cocaine or MDPV, for example, drugs such as amphetamine or methadone, which are dopamine releases, can still continue to cause dopamine release via OCT3, and then that released dopamine is not able to be taken up by the dopamine transporter because we have the DAT blocker in place, perhaps leading to the greater high that is experienced when people take drug combos. So in conclusion then, Dicenium-22 sensitive transporters, putatively OCT3, provide an explanation for the counterintuitive effect of combinations of drugs, releases and blockers. These transporters may also explain, therefore, the therapeutic ineffectiveness of drugs targeting DAT, NET and ZERT for that matter, for the treatment of amphetamine abuse, and that OCT3 may be a novel molecular target for the treatment of psychostimulant abuse and related disorders. So the take-home message from what I've shown you today is don't forget about uptake 2 mechanisms, our organic cation transporter, the plasma membrane monoamine transporter, and the homeostasis of monoaminergic signaling, and mechanisms of drug action in the brain. And just quickly, for anyone who's particularly interested in OCTs in the CNS, there's a new volume of the Handbook of Experimental Pharmacology coming out in 2021. And it has everything you want to know about OCTs in the CNS. And with that, I just want to thank everyone in my lab who helped contribute to these studies, including my collaborators and former lab members, as well as my funding sources. And with that, thank you very much for your attention.
Well, thank you very much, Lynette, for the beautiful presentation. And I've seen that we have 2 questions in the box here below the presentation. Would you like, since they are rather technical questions, perhaps you can try to answer them now that we are fresh from your presentation. Are the experiments done with the Cesium-22 confirmed also by silencing in vitro of OCT3?
Yeah, that's a great question. That's actually something we're doing right now. So I don't have an answer for you, but we are doing both viral knockdown approaches as well as conditional knockdown. So stay tuned. That will be my next presentation.
Okay, and the second question is, how did you determine low and high serotonin concentrations?
Somewhat arbitrarily, I'll say. There was actually a paper published by Butler and colleagues in 1988 where they did a similar experiment but in synaptosomes and found at low concentrations, and they used about 0.5 micromolar serotonin, they found that they could inhibit uptake into the synaptosomes in a selective serotonin reuptake inhibitor sensitive way. And when they increased the concentration to around 2.5 micromolar, it became SSRI insensitive. So we took the concentration that we used from that paper initially, but also taking in mind, the elegant fast-scan cyclic photometry studies from Mark Whiteman's lab, showing the millimolar concentration, like 6 millimolar concentration of serotonin is estimated to be in the synapse. So, we think that even 0.5 micromolar and 2.5 micromolar serotonin might be at the low end of the concentrations that these transporters actually do see in vivo. So the fact that these the activity of uptake 2 transporters are detectable with concentrations of serotonin, as low as 2.5 micromolar is quite impressive, in my opinion, anyway.
Okay, thank you. And now I have a pleasure to introduce Cesare. Cesare Indiveri is a full professor in biochemistry at the University of Calabria in Italy. His research interests cover several aspects of nutrition and metabolism in health and diseases, with particular reference to absorption, distribution, and excretion of nutrients and drugs. Cesare is also involved in drug design, and with his group created a platform for drug transporter interaction testing based on protein expression, purification, and functional assay in vitro ex vivo system. So it's a big pleasure to have you here, and the title of the presentation is the SLC22A4 Alias OCTN1 transporter, novel findings and dark sides.
Thank you, Giuliano, for your kind invitation, and thank you to the MDPI staff for organizing this webinar. Just a few information about my working places are located. That is just in the middle of the Mediterranean Sea, where you can find nice places that hopefully you can reach if the virus will allow this summer. Now going to the topic, OCTN1, as Giuliano has also cited, is a member of a very small subfamily made by 3 members that is part of the larger group of organic cation transporters that indeed is part of the SLC22 family, that includes also anion transporters. These 3 members are present only in 2 vertebrates in line with the N of the acronym that stands for novel, and they are also present in humans. However, the gene for OCTN3 was cancelled in humans. So only the two OCTN1 and OCTN2 are present in humans. Since the first identification of these transporters by the group of Tamai, it was evident that they belong to a carnitin transporter subfamily. But OCTN1 was already clear that it is not or is a very poor carnitin transporter, but it was classified as organic cation transporter on the basis of ability to transport tetraethylammonium, which, however, is not a physiological but a prototype cation. After 20 years and more than 60 papers published on OCTN1 as the main subject, the situation is not much better. OCTN1 has a lot of unknown features so far, and interestingly, the knockout mouse for OCTN1 has no phenotype. What about the searching for physiological substrate of OCTN1? The first report was about the ergothioneine molecule by the group of Grundemann in 2005. They reported ergothioneine as a substrate for OCTN1, but, however, ergothioneine is not a physiological substrate. It is a mushroom metabolite that anyway plays some role in human cells where it cannot be metabolized. Several years after, we proposed another substrate that is acetylcholine, that of course is a more physiological substrate. And years after, the group of Ciarimboli identified OCTN1 as a carrier of nucleoside analogs. Then the group of Cato, by a metabolomic approach, identified spermine as a substrate of OCTN1, and the same group some years later identified homostachydrin, which anyway is still a not physiological substrate, but a plant metabolite. Concerning the transport of ergothioneine that has been studied very deeply, it is transported by a sodium-dependent uptake, and the role of ergothioneine should be defense against oxidative stress, since it is not a physiological compound. Among all the molecules which have been found to be transported by OCTN1, the common feature is the presence of a quaternary ammonium group, or at least a amino group that can be charged at pH, at neutral pH. Thus, we did some bioinformatics to have an idea on how all these compounds can interact with OCTN1, or at least some of them. And we built an homology model of OCTN1 that so far is the most detailed. It has a central core where the substrate should bind, and we found that there are at least 3 residues. A tyrosine residue, which should interact with the ammonium group, forming a cation pigric interaction, and then a hydrophobic residue, phenylalanine, and hydrophilic residue, arginine, that should undergo hydrophobic and hydrophilic interactions with the substrate. As you can see, ergothioneine and acetylcholine are best fitted into this substrate binding sites, while carnitin and acetylcarnitin, that are very poor substrate, are fitted in a worse way. Some in the wild, a Drosophila transporter, which is similar to the transporters of the SLC22 family, was identified. Its name is CG7442. This transporter is a transporter for acetylcholine and choline. And very interestingly, it shares 25% identity with OCTN1. As you can see from a partial alignment of the sequences, OCTN1 and the transporter from Drosophila share the same residues in the substrate binding site, which are not shared with other members of the SLC22 family, apart OCTN2. So this finding still pushed us to continue in our investigation on the role of acetylcholine transport mediated by OCTN1. The first approach was to obtain the recombinant human OCTN1 overexpressed in bacteria and inserting it into proteoliposomes to study the transport in absence of interferences from other transporters. Anyway, to be in a more physiological context, we also use the classical approach, that is adding the radiolabeled acetylcholine to intact cells, and so studying the transport into intact cells. And we also used an intermediate tool, that is extracting membrane protein from intact cells, inserting them into liposomes, obtaining proteoliposomes that in this case carry a protein that is coming from intact cells. By the first approach, that is the recombinant protein, we see that the transport mediated by OCTN1 recognize tetraethylammonium. The same level of transport can be measured with acetylcholine, while both ergothioneine and carnitine can be transported by the recombinant OCTN1, but with a much lower efficiency with respect to acetylcholine. In the attempt to find a more physiological situation in which measured acetylcholine transport, we did this experiment, always with the recombinant protein, in which we studied the transport of acetylcholine in the presence of external sodium. As you can see, the transport is much inhibited by external sodium, so indicating that probably this acetylcholine uptake cannot occur in a physiological environment. Thus, we studied the efflux mode. And we saw that acetylcholine can be exported by means of OCTN1 activity, and interestingly, in this case, external sodium did not affect the transport, as also internal potassium has no effect. So this data indicates that most probably under a physiological condition, the activity of OCTN1 with respect to acetylcholine should be export of acetylcholine, which may be the physiological role of this efflux. We saw that there is a non-neuronal cholinergic system which is active in most human tissues, that is based on the presence of a choline acetyltransferase that can synthesize acetylcholine that is exported and has effects of acetylcholine receptors that are present in all the cells. This non-neuronal cholinergic system has important cell roles, among which the control of inflammation. When we describe the role of OCTN1 in catalyzing acetylcholine efflux, the transporter that was still missing into this non-neuronal cholinergic system was applied to the complete picture, thus explaining how acetylcholine can go out from cells and interact with the acetylcholine receptors present into all these tissues. among which mesothelial cells. So after this, we studied just human mesothelial cells to see whether we can find the same properties of the recombinant protein. Mesothelial cells express OCTN1 at high level, as you can see from this Western blot, in comparison with the recombinant protein, and we see that there is a higher molecular mass due to the presence of glycosylation of the OCTN1 protein. We extracted the protein from mesothelial cells, inserted them into the proteoliposomes, and studied the transport of acetylcholine that is inhibited as found for the recombinant protein by external sodium. While when we measure the efflux, again, it is not inhibited by external sodium nor by internal potassium. Of course, a lot of controls have been performed to be sure that this activity was due to OCTN1 that I will not show in sake of time. We also studied the same transport into intact cells directly, and we again saw that sodium chloride inhibits the transport, and interestingly, there is also efflux of acetylcholine, which is not inhibited by external sodium, confirming our proposal of involvement into the non-neuronal cholinergic system. We also studied the regulation of OCTN1 and found that it is regulated by intracellular ATP in line with the presence of an ATP binding site on the internal phase of the transporter, and we then studied the regulation by cholesterol. If we add cholesterol hemisuccinate, which is a more soluble analog of cholesterol, the transport of acetylcholine and tetraethylammonium is much stimulated, as you can see from these pictures. We studied the same property into intact cells to be sure that this was also a physiological phenomenon, and we, in this case, used methylcyclodextrin, which is a cholesterol sequestering agent, and after controlling that the amount of OCTN1 was not reduced before and after the treatment, we inserted the protein into proteoliposomes and again studied the transport of acetylcholine, and we saw that after cyclodextrin treatment, the transport is much lower. That means that cholesterol indeed is needed for full activity of the transporter. In line with this, OCTN1 share some KARC and CRAC motifs, which are reported as cholesterol binding motifs. And when we see these motifs on the homology model of OCTN1, you see that the motifs are coincident with the docking of 3 molecules of cholesterol that has been performed on the homology model of OCTN1. And as it can be better seen from the top view, the cholesterol molecules face the surface of OCTN1 protein protruding towards the hydrophobic part of the membrane. Then I have to say something on the pathophysiology of acetylcholine transport that we have studied. The only link of OCTN1 with the human pathology is that of the presence of a mutant of OCTN1 that is leucine 503 phenylalanine, which is linked to IBD, and in particular with Crohn's disease. This amino acid residue is located in alpha helix, which corresponds to one of those which interact with the cholesterol molecule. So probably this phenylalanine, which is, of course, which has a steric hindrance higher than the leucine, may cause some impairment on the cholesterol interaction. Anyway, when the mutant was discovered and associated with the Crohn's disease, the group of Taubert, 2005, just in coincidence with the discovery of ergothioneine as substrate of OCTN1, studied the transport of ergothioneine into cells harboring the mutant. In this case, no decrease of transport was observed. Rather, they found an increase of transport of ergothioneine in cells harboring the mutant OCTN1. So this, of course, could not explain the phenotype. Then we studied the transport of acetylcholine catalyzed by the mutant protein, and we had the same results of Taubert. That is, the transport of acetylcholine in the wild-type protein and in the mutant protein is exactly the same. But when we studied the efflux catalyzed by the transporter, we saw that while the wild-type catalyzed the efflux of acetylcholine, the mutant protein is much impaired, thus giving some explanation on the role of OCTN1 and its link with the Crohn's disease. So, altogether, results from our lab and from several other labs can give some conclusion about OCTN1. It is surely a polyspecific transporter, as also Giuliano has highlighted. It catalyzed both uptake and efflux of substrates, and it is regulated besides ATP also by cholesterol. What can we say about the physiological function? We can say that both ergothioneine uptake that should be involved in the defense against oxidative stress and acetylcholine efflux that should be involved into the control of inflammation can play similar roles. Some similar roles can also be played by other substrates that may be involved with defense against oxidative stress or control of inflammation, as you have seen from the type of substrate transported. However, there are a lot of dark sides still on OCTN1. First of all, the molecular basis of polyspecificity. We still do not know how this can be explained. The molecular mechanism of efflux, while the transport mode of ergothioneine is a sodium-dependent uptake, the molecular mechanism of efflux is still not known. A striking question is how sodium can be stimulator of ergothioneine uptake but inhibitor of acetylcholine uptake. Another dark side is the association between ergothioneine uptake and IBD. And of course, as for many other transporters, a big dark side is the 3D structure that, when should be solved, will give a lot of answers to several of these dark sides. At the end, I want to thank all the participants to the webinar and my co-workers, Lorena, Michele, Maria Francesca, and Lara, who are highlighted in this picture that contributed to the big work on OCTN1. Thank you.
Thank you, Cesare, for the excellent presentation. I have a question for you. Did you try to perform acetylcholine efflux experiment in the presence of ergothioneine outside of cells?
Yes, we tried to perform both acetylcholine uptake and acetylcholine efflux in the presence of ergothioneine, but we did not see any variations.
Mm-hmm. So it stays a mystery.
Yeah, a big mystery.
Yeah. Okay. So thank you very much again. And now I'm happy to introduce Michele Visentin. He's born in Venice, in Italy. And he has a PhD in biochemistry and pharmacology from the University of Udine in Italy. And his interest in the study of membrane transporter began after joining as a visiting graduate student the research group of David Goldman at the Albert Einstein Medical College in New York. Now, Dr. Visentin is a group leader in the Department of Clinical Pharmacology and Toxicology of the University Hospital of Zurich, where his main focus is elucidating novel biochemical features of organic cation transporters and their pathological and pharmacological ramifications. The title of his presentation is Organic Cation Transporters in Pathology, Imaging, and Current Therapy of Cancer.
Thank you, Giuliano. I think you can see my screen.
Yes. Yes.
Perfect. And I hope you can hear me well. Thanks to the NDPI team and to all the people that are attending the webinar. So, well, you guys make my life quite easier because you have a really, really nice introduction on the biochemistry and the mechanism of transport of most of the organic cation transporter. So I actually cut the presentation more on a really on a clinical level, focusing on cancer, this complicated disease, multifaceted, that we can try to describe, if we have to use a few words, as a genomic instability situation with epigenetic alteration that lead to a phenotype characterized by uncontrolled cell proliferation, defective apoptosis, acquisition of invasion properties, ability to escape the immune system, and huge change in the energy metabolism. All situations that affect diagnosis and therapy of the disease, but on the other hand, that we are trying to exploit to for diagnostic and therapeutic aspect. So in this highly simplified frame, where do we place OCTs? How OCTs can the role played by the OCTs in the diagnosis and the therapy of cancer. And OCTs, as you guys mentioned, transport a lot of metabolites. And as we said, metabolites are the supply of metabolites in cancer is heavily changed. And this is something that now is in the diagnostic routine, measuring, assessing the metabolic activity of cancer by positron emission tomography, in which basically a radio-labeled metabolite is injected, and then a snapshot of the activity of the normal and the cancer tissues can be taken. The research is ongoing to develop new radio tracers, but in principle, in clinical routine, there are two main metabolites: the glucose, which is usually cancer cells are avid for glucose since the anaerobic glycolysis is particularly boosted in cancer cells, The other is choline, which is the precursor of phosphatidylcholine, which is one of the main lipid species that constitute the cell membranes. Now, what in radiopharmaceutical we do is we can label a carbon of a glucose or a carbon of a choline as 11C with the 11C radioisotope, or more commonly, we modify the molecule by adding a fluorine, and then we label the fluorine as 18F. The advantage is particularly is practical. There is an increase by adding the fluorine, by labeling the fluorine, we can increase the half-life of the radioisotope from 20 minutes to almost 2 hours. And this, of course, is very practical because usually the site of synthesis of the radio tracer is different from the site where the exam takes place. So, of course, I'm going to focus on now fluorocholine and choline because, as mentioned before, choline is to some extent a substrate of OCT. It is particularly a very good substrate of the human OCT2. Here you can see how the behavior of the transport of choline by human OCT2 as a function of the extracellular concentration of substrate, and you can see a sigmoidal behavior, which suggests an allosteric binding, and this is confirmed when we do a linear transformation, which is supposed to be linear in case of a one binding site kinetics, but then becomes a curve that is concave towards the y-axis, indicating a positive cooperativity. So the binding of the first molecule stimulate the binding and facilitate the binding of the second molecule. It is likely that OCT2 is the main contributor of the handling of choline in the kidney, as you can see here from this is a staining of normal kidney, and you can see the clear, intense basolateral staining for OCT2, confirming the high expression of this transporter at this level. What was interesting was that there were a couple of papers showing how choline, in this case 11C choline, can be particularly helpful in the follow-up, in the staging and the follow-up of renal cancer, especially to flag long-distance metastasis. This is a patient, a head-to-head comparison, basically between a choline PET and a fluorodeoxyglucose PET of a patient that underwent a nephrectomy, and that after years, basically during the follow-up, was found with a number of long-distance metastases, or you see skin, clavicular metastases, that were actually not flagged at all by the fluorodeoxyglucose. Plus minus at the same time, in another, this is a case report of a patient, again, diagnosed with clear cell renal carcinoma, underwent a nephrectomy, and then after a while, during the follow-up, pancreatic metastasis was flagged with the 11C choline, but you can see there is basically no signal with the 18 fluorodeoxyglucose. So this to us was quite interesting because at the same time we were able to study the expression of OCT2 in a tissue microarray of clear cell renal carcinoma. So we analyzed about 300 samples, and we found basically no OCT2 in the clear cell renal carcinoma. However, and this was also confirmed by others, however, when we were able to analyze, at least at the mRNA level, of OCT2 in the primary tumor and in the metastatic tumor, we kind of saw that OCT2 was switched on again in the metastatic tissue, which of course makes sense and provide an explanation why OCT2, which is a very good subset of choline, can flag metastatic cancer, renal cancer. Then the question is, so far we talk about 11C choline, but what happened when we switch from choline to fluorocholine? Well, in principle, the fluorination is a pretty heavy change in the molecule. And because fluorine is the most electronegative element, so when it interacts with the carbon, basically is pulling the pairs of electrons, and this changes the electronegativity of the neighboring carbons. And this, to the overall molecule, can have an effect on the protonation, of the overall protonation of the molecule, and on the lipophilicity of the molecule. In fact, actually, there are evidence that if we incorporate fluorinated amino acid into proteins, we can change the thermostability, the proteolytic stability, also binding affinity and enzymatic, so pretty much everything of the protein. And this is actually quite true also when we are looking at the interaction with transporter, in this case with the OCTs. Again, as I showed you before, this is the typical, this is the AD-OFT plot for the kinetic of choline, so the homotropic positive cooperativity. And when we measure the kinetics of fluorocholine mediated by the human OCT2, we have a clearly different behavior. And what you can see is that you can clearly define two regression lines, which means two slopes, which again in the AD-OFT plot reflect two independent Km, one at the high affinity, and so a high affinity binding site, and a low affinity binding site. That two binding sites that, unlike for the choline, are most likely independent one to the other. So this was interesting also then, because it's interesting because fluorocholine seems to become also a better substrate of other organic cation, like organic cation transporter 1 and OCT3. This is just a time course of the transport of fluorocholine by OCT1 and OCT3 overexpressing cells, and this to me is particularly interesting when then I try to explain some imaging, so the pattern of imaging of fluorocholine in the hepatic tissue. This is a relatively old study, again, head-to-head comparison with fluorocholine PET and fluorodeoxyglucose PET for a patient that was treated for hepatocellular carcinoma. And at the follow-up, you can see how fluorocholine is much more effective in flagging the relapse of the cancer in comparison with fluorodeoxyglucose. Another interesting pattern was shown by a group in Amsterdam. These are two benign hepatic lesions, focal nodular hyperplasia and hepatocellular adenoma. This is a standard CT, and you can see a similar pattern, so a brighter signal here and here, so quite indistinguishable one to another. When a fluorocholine PET is coupled to the CT, we have a clearly different pattern, where basically the focal nodular hyperplasia is brighter, is avid, very avid for fluorocholine, whereas the adenoma is again almost indistinguishable or even cold compared with the normal tissue. And this is quite interesting because in some cases, if you cannot distinguish at the imaging the FNH to the HCA, you need to go for a biopsy or doing a partial hepatectomy that, of course, can be avoided if one is sure that of a focal nodular hyperplasia, since is a relatively benign, is completely benign, and whereas the HCA in certain cases can transform into an hepatocellular carcinoma. And we try to explain, we hypothesize that the expression of OCT1 and OCT3, which are the two main OCTs in the liver, both expressed at the sinusoidal side, can affect this uptake. We had the possibility, in collaboration with, again, with the group of Amsterdam, of Thomas van Gool, to obtain the normal and the cancer tissue from 4 patients diagnosed with HCA, and of which we had available the PET-CT imaging. So in general, you can see from with the white arrow the area of the HCA. In the first case, is clearly hypointense. Also here is relatively hypointense. A clear hypointensity is also here in case number 3. For case number 4, we have basically is not visible, we can say is iso-intense in comparison with the normal tissue. And when we analyze the expression of OCT1 and OCT3 and quantify the intensity of the signal relative to the normal tissue, we can see there is a certain consistency between the OCT1, OCT3 expression and the PET signal, with kind of opposite expression trend in the case number 4 between OCT3 and OCT1 that may explain the iso intensity of the 4th case. So along this line, that tried to figure out how we can, what is the role of the OCTs in cancer. We recently found, and this is still, let's say, unpublished data, hopefully published soon, we found that OCTN2, among all the OCTs, is overexpressed in esophageal cancer. This study was done in collaboration with Shandong University in China. Esophageal cancer has 2 main histotypes, squamous cell carcinoma and adenocarcinoma, with the squamous cell carcinoma particularly frequent in the Asian population, and the adenocarcinoma more frequent in the Caucasian population. So we combined basically 2 cohorts, and we found a general overexpression of OCTN2. This is basically just a group of patients in which tissue was available also for Western blot. And we were able, for some of these, especially those that underwent partial esophagectomy, to culture the cells This is a staining of OCTN2 in the primary cells derived from patients, primary esophageal cancer cells derived from patients. And we were able to assess the functionality of OCTN2. So we measure the transport of carnitine in the presence or absence of sodium, since the transport of carnitine mediated by OCTN2 is strictly sodium dependent. And we could also group the two transporter, this number of cell line derived from patient in low expressing OCTN2 and high expressing OCTN2. And when these cells were exposed to oxaliplatin, which has been shown to be a substrate of OCTN2, we found that basically the cells that express a higher level of OCTN2 are more sensitive than this is just a measurement of the ATP, cellular ATP as a readout, are more sensitive to oxaliplatin. Oxaliplatin is often used in the pharmacological treatment of esophageal cancer, as often in combination with irinotecan or 5-fluorouracil. And we had the possibility to study a cohort of more than 60 patients that underwent surgery and then were treated with oxaliplatin-based therapy. We followed them up for 3 years. And what we found is that when we group them in high expression, this was measured by histology, the expression quantified by histology of the high expressing group is basically has better outcome in terms of progression-free survival and in terms of overall survival, which with an HR, with an hazard ratio that is plus minus 3 times. So this is probably, we think this is related to the ability of these cells to incorporate more oxaliplatin. And this is particularly interesting because it is also done on a decent number of patients, all with locally advanced esophageal carcinoma. So far, we basically consider only the expression as a determinant of OCT, of transporter activity, and as a determinant of accumulation of the radiotracer or of the drug. But it's not that simple, and this is a little bit actually what already Cesare introduced. Transporters, of course, need to interact with the lipid bilayer, and we know that cholesterol is particularly relevant in the regulation of the function of several transmembrane protein, from a signaling protein to, of course, transporters. And we know that this can be relevant also in cancer. And why? Because metastatic cancer cells have been shown to have relatively low cholesterol content, which of course increase the flexibility of the membrane and may facilitate the invasion process. And in multi-drug resistant cancer cells, so cells that become or are resistant to multiple drugs, have high cholesterol content at the membrane level. And this is probably because it lowers the permeability, especially to lipophilic drugs. But so it is also important to consider how this component can affect the function of the transporter, also the organic cation transporters. And we did again go back and study OCT 2, we use a similar approach that Cesare described. So we use methyl beta cyclodextrin, which is oligosaccharide with a torus-shaped oligosaccharide with an hydrophobic task, which seems to be particularly selective for cholesterol. So when the cells are short time exposed to MBCD, we are able to remove, in this case, up to 50% of the cholesterol content. This is a 20-minute incubation with MBCD, and you can see on the right side that the level of the other main lipid species from sphingomyelin, phosphatidylcholine, phosphatidylserine, inositol, and ethanolamine seems to be relatively stable. And when we do that, we see that this is again the kinetics of MPP, in this case MPP as a prototypical substrate for OCT2 with or without the treatment with MBCD. And you can see there is a relatively quick saturation. And when we actually look at the linear transformation, again, we see an allosteric binding in the control cells, but we see a pretty nice straight line in the presence of MBCD. So our conclusion here is that really the reduction of cholesterol is also quite important for to preserve the allosterism of the human OCT2. And in a similar fashion, we studied also OCTN2. And again, here we see an induction of the transport when cholesterol is present, and we were able to confirm this also with the help of Cesare in proteoliposomes. So by increasing the amount of cholesterol, we were able to induce the transport of carnitine. And I think with this, I'm done. This is a little summary. So I want to conclude by saying that OCT expression level in tumors depends, of course, on individual characteristic, but also on the histotype in certain case and on the location, as we saw from for the renal cancer between primary tumor and distant metastasis. I hope I convinced you that OCTs may be quite relevant in the accumulation of the PET tracer fluorocholine in cancer cells, and that OCTN2 plays a major role in the accumulation of oxaliplatin in esophageal cancer, also in vivo probably. And finally, that expression is not the only thing that matters to predict the function of the transporter in cancer. And of course, the lipid microenvironment, which is quite dynamic in cancer, brings an additional level of variability to the function of OCT in cancer. And I just leave here, maybe this can be good for the final discussion. What are the next question? Of course, what is the meaning, the biological meaning of the OCT2 modulation in renal cancer, both in the primary and in the distant metastasis? And of course, whether we can some sort of hijack OCT, so develop rationally designed radio tracer that can couple the diagnostic performance and the therapeutic guidance. So in other words, can also the imaging give us a clue of whether a certain drug can be useful for the treatment of a certain cancer of a certain individual. And then again, what is the role of lipid OCT interaction in drug resistance?
Thank you very much, Michele, also for the presentation. Impressive data. And there is already a question for you in the F&A box. So you can see, do you think that the overexpression of OCTN2 in cancer cells has to do with metabolic changes occurring at the level of lipid metabolism in cancer cell, and then may have to do with carnitine requirement?
It's possible. It would have been also actually, we tried to also figure out whether these primary cells have some sort of, show also some sort of difference in terms of cell proliferation, which we didn't find. But I think the question is absolutely spot on, considering also that the last couple of years ago, a paper was published by a group in Greifswald showing that indeed the OCTN2 expression was associated with a more aggressive, for instance, phenotype of glioblastoma. And in that case, it was not related to the treatment, but it was at the end basically suggested that this, of course, has probably something to do with the role of carnitine and the overall role of carnitine in lipid and lipid metabolism, with the aggressiveness and invasiveness of cancer. So it could well be, but we don't know for sure.
Okay. I have also a question. Also, I wonder whether you tested whether fluorocholine is the substrate of OATs, organic ion transporters.
No, we haven't tested that. We tested all the OCT1 to 3 and also OCTN1 and OCTN2, but we haven't looked at the OAT.
Mm. Yeah, because of a negative charge in fluorides. So there are a couple of questions still in the box, but they are for me, I think. There was a question whether post-translational modification might affect substrate specificity of OCTs and MAIDs. Yes, so we investigated this point in ret organic cation transporter 1, and there we found that phosphorylation by PKC change the affinity of the transporter, also increases the affinity of the transporter, so modify the function. But this is specific for this transporter. When we try the same approach using HOCT1 or HOCT2, we could not observe any effect, also any stimulation, any change in affinity, I think. And I hope that I could answer the question. And Lorena asked whether ergothioneine can interact with OCT in polarized cells. I don't know it. Also, I'm not aware of any data reporting something like that. So I don't see any further question in the box. And I see Mina.
Yes, sorry, if I may, I see a raised hand, Oscar Zanu, if you want to.
No, I don't have any question. I would like to thank presenters. It is a great opportunity to follow you, to listen to you, and to learn from you. Thank you very much.
So we have still a little bit time for discussion. Mina, is that right?
Giuliano, can I have just a quick question? I guess for you and Lynette, OCT3, what is known about OCT3, but in general, the OCT in the human brain.
Yeah.
So you extensively show convincing data from you from other lab about OCTs in rodents, but is there anything that you can share with us?
Yeah, I mean, the expression of the OCTs and PMAT in human brain, excuse me, is not all that well known yet. They're all there, but it's difficult to know at this stage how they compare with rodents. So, I mean, there's differences between expression in mice versus rats. So I think it's reasonable to presume that there's going to be differences in human as well. So until such time as we, one of the big things hamstringing us in these studies is that there are no selective ligands available for the 3 OCT isoforms and PMAT. So we're kind of stuck with Desinium-22 right now, which makes it really difficult to look at interspecies differences as well. We are working with medicinal chemists right now to try and get selective compounds, but I think, as you say, I mean, what we've found, we and others have found in mice and rats, OCT3 really seems to be the transporter, at least for these psychotherapeutic effects. It's not to say, though, in humans, perhaps it might be the plasma membrane monoamine transporter. We'll see as the research goes, but I think what we're learning in rodents will translate to humans. We might just find that in humans it's not OCT3, it might be one of the other isoforms or PMAT.
Do you think it's more to try to have a thorough idea of what's going on in the human tissue? Is it more an issue of course, practical issue of ethical issues, or technical, rather, for instance, having good antibodies, selecting the specific antibody for the human OCT3 that can help them in trying to elucidate a little bit?
Yeah, there are satisfactory antibodies around. I'd love to get my hands on a human brain bank. That'd be great. We could run some of our own analyses. But as far as, any radio tracer kind of studies go, we don't have the tools right now.
I have also a question in this direction. When we think of addressing the OCT3 in the brain with drugs, how can these drugs cross the blood-brain barrier? Do you know whether there are also OCTs expressed that can do it, or other transport systems?
So the blood-brain barrier and how drugs are getting in via the organic cation transporters, yeah, they're obviously very important for that. And the plasma membrane monoamine transporter and OCT2, I believe I need my colleague Joanne Wang here. She's the true expert on blood-brain barrier and choroid plexus expression of these transporters. But I believe it's OCT2 and PMAT are the most important in drug transport. But OCT1 and OCT3 are also expressed there as well.
Okay. So I would like to ask a question for Cesare also. Did you measure whether the transport by OCTN1 is pH dependent?
Yes, it is. And the transport increases at a light alkaline pH, at pH 8. Then it decreases at a higher pH. Anyway, there may be some effects by buffers, because we see that, for example, Tris buffer is an inhibitor of OCTN1 of acetylcholine uptake, for example, because it is at acidic pH, it is charged, so it behaves as like sodium. Thus, we cannot say that the effects seen can be all attributed to a sensitivity of the transporter to pH or to other effects that may be mediated by some ions, which are, while the efflux is not dependent by pH.
Yeah, it's very interesting that this acetylcholine transport could be relevant in inflammatory conditions. No?
Yes, I have seen your theory that these transporters may have a role in an interorgan signaling. So this is very, very in agreement with a signaling role for acetylcholine, or another substrate that is well transported by OCTN1 is choline.
Mm-hmm.
And I have seen the slides of Michele. I have a question that is, have you found any variation of OCTN1 among tumor cells and normal cells?
I have to go back and check, but we publish all this data in the same paper, and we can discuss that. We have, for the OCTN1, we just look at the mRNA, but for in the liver we have this data. For the I think you refer more to the liver data, so the data related to the PET that I show. So in the kidney we haven't checked at all. In the kidney tissues, in the liver we check the mRNA of OCTN1, but I don't recall much of a difference. But we can take a closer look.
So I see that we are over the time, also already 5 minutes. And my question for Mina is, should we close the session now? Or?
Yes, if there are no more questions.
There are no more questions. I have only 2 slides to close this webinar. Then, as here, I think the field of transporter research is still very interesting. Still, there are many questions that have to be answered, and so I hope that more people are now motivated to submit work for the special issue. And I would recall that the deadline for manuscript submission is at the end of October 2021. And I would like also to mention that there is an interesting conference where Cesare, Michele, and me met already. The transporter Tage, also transport days, also in Greifswald. And this year, because of Corona pandemic, this conference is via Zoom. So also people from the US or the other part of the world that are very far from Germany can participate when they will. And so here is only an information, and you can see we have shown a couple of keynote speakers. And when you want, you can ask me for more information about this conference. And so now I am ready. I would like to thank,
Yes, go ahead.
I would like to thank you all for the participation, for the very interesting presentations. And I hope we have the chance to work together and probably I have many questions for Lynette for the future. And with Cesare and Michele, we are working already together with a couple of projects. And so I would like to close here the webinar, and I thank you all the participants also for the questions also.
So I do see a raised hand just before we close the webinar. That's why I thought maybe you wanted to see the question.
Yeah.
It's Johannes Hagos.
Johannes.
Hi.
Hi.
Hi. Yeah, thank you very much for everybody. To people who don't know me, Johannes Hagos from Porta Celtec. And I have just one question to Cesare about these liposomes and OCT1. Have you ever tested a higher potassium concentration on the outside of the lipo? Because if you give sodium, it is blocked. Yeah, if I if I understood it right.
Yes.
But
yes.
what is if you give a higher concentration of potassium in extravesicular or extra liposomal?
Yeah, it's the same. Potassium has the same inhibitory effect of sodium on the external side, while on the internal side, it has no effect.
Because it could be also some role of potassium in vivo that we can see in vivo with a high potassium concentration in the cell, maybe will drive the efflux of acetylcholine in a vivo situation.
Yes.
That's why
yes.
I was interested
Yes.
that the higher concentration we cannot avoid, because you do have the higher concentrations, and that could be maybe a mechanism. But if you already tested it outside, then it's not maybe involved.
Yeah, yeah. Inside it has no effect. Outside it inhibits like sodium.
Okay. Okay. Good. Yeah. Thank you very much.
You're welcome.
Now we can close the webinar.
Yes, I want to thank you, the chair and the speakers, for their wonderful talks. I hope, and I thank the attendees, both people who joined us here at webinar and those who watched us live. I hope you all enjoyed the webinar, and I hope that we can see you all soon again. As a reminder, you can access the recording on the webinar's webpage as soon as possible. Thank you so very much. I wish you all a happy day or evening based on where you are in the world. Thank you. Bye-bye, everyone.
Bye-bye.
Bye. Thank you very much.
Thank you. Bye.